Welcome to another episode of the Med Madness podcast, where we dive deep into the intricacies of medical science to help you ace your USMLE exams. Today, we're focusing on a crucial aspect of endocrinology, the intracellular signaling pathway of cortisol. Understanding cortisol's mechanism of action is vital for comprehending how our body manages stress, metabolism, and immune responses. Next, let's discuss cortisol's primary role. Cortisol is a steroid hormone that is essential for regulating metabolism, immune response, and our body's stress response. During periods of prolonged fasting or stress, cortisol plays a pivotal role in maintaining blood glucose levels by increasing the transcription of enzymes involved in gluconeogenesis, lipolysis, and proteolysis. Now let's explore the mechanism of cortisol action. Initially, intracellular cortisol receptors are found in the cytoplasm associated with heat shock proteins, which keep them in an inactive state. When cortisol binds to the carboxy-terminal portion of the receptor, it causes the release of the heat shock proteins, allowing the receptor to dimerize. Next, the cortisol receptor complexes dimerize, forming activated homodimers. These activated homodimers are then transported to the nucleus. Inside the nucleus, the activated homodimers bind to hormone-responsive DNA elements in the promoter region of target genes. This binding increases the transcription of specific genes, leading to the production of enzymes necessary for gluconeogenesis, which is generating glucose from non-carbohydrate sources, lipolysis, which is breaking down lipids to release fatty acids, and proteolysis, which is breaking down proteins to release amino acids. To put this into perspective, let's consider a real-life case study. Imagine a patient named Jane who experienced a prolonged fast. Despite not eating for an extended period, Jane's blood glucose levels remained stable. This stability is due to the suppression of insulin secretion and the activation of counter-regulatory hormones like glucagon and cortisol. Cortisol, by binding to its intracellular receptors and altering gene expression, ensures that Jane's body can produce glucose through gluconeogenesis, breaking down fats and proteins to maintain her energy levels. Moving on, let's compare cortisol's mechanism with other hormones. Catecholamines such as epinephrine and norepinephrine and glucagon bind to extracellular, transmembrane, G-protein-coupled receptors, activating adenyl cyclase and increasing cyclic AMP production. Growth hormone acts via an extracellular transmembrane receptor that activates the JAK-STAT pathway. Insulin acts on an extracellular transmembrane receptor with intrinsic tyrosine kinase activity. To conclude, cortisol is a steroid hormone that binds to intracellular receptors, leading to changes in gene expression. It plays a vital role in increasing the transcription of enzymes necessary for gluconeogenesis, lipolysis, and proteolysis, which are essential processes during stress and prolonged fasting. We hope this episode has provided you with a clear understanding of cortisol's intracellular signaling pathway. To reinforce your learning, don't forget to engage with our pre- and post-podcast quizzes and Anki flashcards. These tools are designed to help you retain key concepts and excel in your exams. Thank you for tuning in to Med Madness. Stay curious, stay focused, and let's ace those exams together.